updated notes as per the latest syllabus and MCQs, beginning from the beginners level, going up to the standard multiple choice questions. So you can assess your level based on the MCQs. Apart from that, we have personalized counseling sessions. You can anytime book a slot with us if you need any extra guidance apart from academic support. Now, there are 3 different subscription plans at Auricle, starting from the basic, which is for 3 months and will cost you around 15K. Then you have standard plan, which is for 6 months and it is for 30K. And then you have premium plan, which is for 12 months and will cost you 50K. And apart from that, you can also get additional 20% off if you use my promo code as displayed on the screen. Okay. So let's begin with the class. Taking up a clinical case, a 24-year-old woman presents with diarrhea, dysphagia, jaundice, and white transverse lines in the fingernails. The patient is diagnosed with arsenic poisoning. Okay. Which of the following enzymes? So this is a very direct question. It is a case of inhibition of the cycle. So once you're done with TCA, you will know what are the arsenic poisoning effects and which enzyme it will be affecting. Okay. So now let's begin. Your TCA cycle, fully known as tricarboxylic acid cycle, it is so known because one of the intermediates in this cycle is a tricarboxylic acid, which is your citrate. Okay. So on the name of the citrate, it is also known as citric acid cycle. So it is known as tricarboxylic acid cycle because a tricarboxylic acid is involved. And what is that? That is your citric acid, and hence the name citric acid as well. Okay, so it has 3 names: tricarboxylic acid, citric acid, and Krebs cycle. Krebs cycle because it is based on the person who first elucidated the cycle. That is your Hans Krebs. Okay, and remember, urea cycle was known as Krebs-Henseleit cycle. So don't get confused between the 2 cycles. TCA is Krebs cycle and urea cycle is Krebs-Henseleit. Okay. Now beginning with the concept of TCA cycle, that when and how does TCA take place? So TCA is the final common oxidative pathway for macronutrients. Okay, so what does it mean? That all the macronutrients that you have through diet, that is your carbohydrates, lipid, proteins, they are all ultimately going to form acetyl-CoA, and that acetyl-CoA is going to get oxidized via TCA. Therefore, we say it is the final and common and oxidative. 3 important words, okay? Final, common, and oxidative, because ultimately the complete oxidation of whatever you had in the diet of all the macronutrients is going to happen via TCA. And this is common for all the macronutrients. And this is an oxidative pathway. So therefore, TCA is known as the final common oxidative pathway for macronutrients that you intake. Now, all the macronutrients form an intermediate, which is acetyl-CoA. And this acetyl-CoA will enter TCA. And in the TCA, the acetyl-CoA is completely oxidized. And what is generated? Reducing equivalents are generated. So when something is getting oxidized, some other thing has to get produced. Okay. So here, when your acetyl-CoA is oxidized, something will be reduced. So therefore, what is being generated? Your reducing equivalents are being generated. They are getting reduced. And then those reducing equivalents, they will be entering electron transport chain. And in the electron transport chain, what happens? Your ATP is generated. Okay. So I'll give you a little concept map. See, your carbohydrate. What happens to the carbohydrate that you intake? it undergoes glycolysis, okay? And in the glycolysis, what is formed? Pyruvate is formed, okay? Now your proteins are broken down into amino acids, and your ketogenic amino acids, when they catabolize, they also end up forming what? Acetyl-CoA. And then you have lipids. Lipids also we've seen, they give fatty acid on beta lipids. The fatty acid in the lipids on beta oxidation, it also gives what? Acetyl-CoA. And your carbohydrate, after glycolysis, they form pyruvate, and this pyruvate will undergo link reaction, and link reaction is carried out by your pyruvate dehydrogenase enzyme. And what does it form? It also forms acetyl-CoA. So you see, your carbohydrates, they will form pyruvate via glycolysis, and then that pyruvate via link reaction. Link reaction is the link between your TCA and glycolysis. Okay, because this pyruvate is coming from glycolysis, okay? And this acetyl-CoA that is being formed will be entering TCA. So your link reaction is the link between, you can see, it is linking what? It is linking glycolysis, and this will enter TCA. So it is linking glycolysis and TCA. That is your link reaction. So that is how your carbohydrates ultimately add on to the acetyl-CoA pool in the body. Then your protein, your ketogenic protein, we've already studied that we had glucogenic and ketogenic protein. So your ketogenic protein on catabolism, you've seen that they form acetyl-CoA. That is the reason we are taking TCA after we are done with the metabolism of all these macronutrients. Lipids also, we've already studied that how the fatty acids, they undergo beta oxidation and they ultimately end up forming acetyl-CoA. So you see all your macronutrients, ultimately, they converge into acetyl-CoA. And now this acetyl-CoA will enter TCA. And in the TCA, this acetyl-CoA, which is a 2-carbon compound, this will get completely oxidized. So when something is getting oxidized, something has to be reduced. So the oxidation of acetyl-CoA, it leads to the generation of reducing equivalents. Now those reducing equivalents, they enter electron transport chain and lead to the production of ATP. So that was about the concept of like TCA. Okay. Now let us have a look at the site. So it takes place in all organs with mitochondria, and the organelle is your mitochondrial matrix. So all enzymes of the TCA are present in the mitochondrial matrix except succinate dehydrogenase. Okay. So your succinate dehydrogenase is seen as a part of electron transport chain as well. In electron transport chain, when we will study, you will see that it is composed of different kind of complexes. So the complex 2 of electron transport chain, it has succinate dehydrogenase, and it is present in the inner mitochondrial membrane. So sometimes you might get an MCQ where it says which of the following TCA enzymes are not present in the mitochondrial matrix, and you will have multiple options, out of which you will have to mark succinate dehydrogenase because it is not present in the matrix. Instead, it is present in the inner mitochondrial membrane because it is seen as a part of electron transport chain as complex 2 of ETC. Okay. Now, before we take the individual steps of TCA, let me give you an overview of TCA. Okay. So your TCA, it can be divided into 2 halves. The first half is involved in the oxidation of acetyl-CoA, and the second half is involved in the regeneration of oxaloacetate. See, let's divide this into 2 halves. Okay. So this will be the first half. In the first half, what is happening? The acetyl-CoA is completely oxidized. Okay. And this is the oxidation will take place how? Via decarboxylation reactions. Okay. So your carbon will be lost as carbon dioxide. Two carbon dioxide molecules will be liberated. So you can see 1 and 2. So what is happening? Your 2 carbon acetyl CoA combines with a 3 carbon oxaloacetate to form a 6 carbon citrate. Okay. Now, see, this is what it says as well. 2 carbon acetyl CoA combines with 4 carbon oxaloacetate to form a 6 carbon citrate. The 2 carbons of acetyl CoA are completely oxidized and carbon dioxide is produced via decarboxylation reactions. So you see 1 carbon dioxide removed here, 1 carbon dioxide removed here, but you see what is also produced, NADH are also produced. So this is not a simple decarboxylation reaction. Instead, it is oxidative decarboxylations. Okay. So there is oxidative decarboxylation taking place. So your 6 carbon citrate has been after decarboxylation, 2 carbon dioxide being removed, it has been converted into a 4 carbon succinyl CoA. This here. Okay. So that was about the first half of your electron transport chain. Now this is the second half. In the second half, what is happening? It is regeneration of oxaloacetate. So you see this 4 carbon acetyl succinyl CoA, 4 carbon succinyl CoA changes into 4 carbon oxaloacetate. Okay, so your oxaloacetate, which was entering the TCA, has been regenerated. You see, so we can say that oxaloacetate is considered to have a catalytic role. Okay, it is acting as a catalyst because it enters TCA and it comes out unchanged just like a catalyst. So only a small quantity of oxaloacetate is needed to oxidize large quantity of acetyl CoA. Since we can say, we can consider that our oxaloacetate is having a catalytic role. So a small quantity of oxaloacetate can oxidize large quantities of acetyl CoA. So that was about the overview of TCA cycle. And we say that this is a cycle and not a pathway because your oxaloacetate here is regenerated. Okay, therefore it is not a pathway but a cycle. You see, it is taking place in a cyclic manner. Oxaloacetate is being regenerated just like we have studied urea cycle. That is also a cycle. It is not a pathway because there also the substrate is being regenerated and here also your oxaloacetate is being regenerated. So that was the overview of TCA cycle. Let me just repeat it. What is happening? What happens in TCA cycle? Your 2 carbon acetyl CoA combines with 4 carbon oxaloacetate to form 6 carbon citrate. Now from that 6 carbon citrate, it undergoes oxidative decarboxylation and subsequently 2 carbon dioxides are released. Okay. So what do you end up after 2 carbon dioxide have been released? That is, you lose 2 carbon dioxides. So your 6 carbon changes into a 4 carbon compound, that is succinyl CoA. Now this succinyl CoA, it undergoes subsequent reactions to ultimately form another 4-carbon compound, which is your oxaloacetate. So we say that our oxaloacetate has been regenerated, and therefore it is considered to have a catalytic role in TCA because it enters TCA and is regenerated, it comes out unchanged. So that was briefly about TCA. Now let's take the steps of TCA in detail. So oxaloacetate, it combines with acetyl-CoA to form citrate. This is the first step. And oxaloacetate is considered as the first substrate. Now, why is oxaloacetate considered the first substrate and not acetyl-CoA? Because we're saying oxaloacetate and acetyl-CoA are combining. So I'll just tell you why. It is considered as the first substrate because the enzyme, that is your citrate synthase, is already bound to oxaloacetate, and then to that acetyl-CoA is attaching. Okay, so let's consider that this is your, okay, let's consider this is your enzyme citrate synthase. Now it's saying that your oxaloacetate is considered as the first substrate because to your enzyme citrate synthase, oxaloacetate is already bound to the enzyme, which is citrate synthase here, and then to this, what is happening? Your acetyl-CoA is attaching. So, if the MCQ, the question comes like, what is the first substrate of TCA? And then your options are oxaloacetate and acetyl-CoA, the rest of the options. So, your answer will be oxaloacetate. Oxaloacetate is considered as the first substrate of TCA. And the enzyme of this reaction is citrate synthase. This is an irreversible and regulatory step of the cycle. It is a reversible and regulatory step. Now, citrate can enter TCA or fatty acid synthesis. In fatty acid synthesis, we've already seen how citrate was forming and then it was coming out as citrate, and then again via citrate lyase enzyme, it was being broken down. So if the body needs fatty acid, so the citrate would enter fatty acid synthesis. Okay. Now the second step, citrate to isocitrate. This is brought about by the enzyme ecotinase, which is also a lyase enzyme, like the previous enzyme. Now this is taking place in a two-steps process. Initially, there is dehydration and citrate converts to cis-ecotinate. Then there is rehydration and cis-ecotinate converts to isocitrate. So it is saying that this is a two-step process where your citrate first undergoes dehydration, that is removal of water. So water molecule is removed and what is formed? Cis-aconitate. And then rehydration takes place, water is added and what is formed? Isocitrate is formed. Okay. So this is what it is saying. Now, equinase, it is an iron-containing enzyme. And it is a moonlighting enzyme. Now, what are moonlighting enzymes? They are those enzymes which, apart from enzymatic activity, has some other action as well. Usually, your enzymes only have one role, that is to catalyze reactions. Okay, they have only enzymatic activity, catalysis. But moonlighting enzymes, apart from the enzymatic activity, they have some other function in the body as well. So your equinase is one such enzyme. It is a moonlighting enzyme. So the enzymatic action of equinase is conversion of citrate into isocitrate. So that is its enzymatic action, to convert citrate into isocitrate. And the other activity of equinase is it is involved in iron homeostasis, that is regulation of normal level of iron in the body. So you should remember this. Let us have a look here in this cycle. Cu2 carbon acetyl-CoA combines with 4 carbon oxaloacetate to form your 6 carbon citrate. Okay, and the enzyme is citrate synthase. Okay, now what is happening? Your citrate, it is acted upon by aconitase, and you can see two steps. Firstly, water is removed, that is dehydration, and then you can see water is being added, rehydration, and what is being formed? Isocitrate is being formed. Okay, now let's see. Before that, let's take an MCQ. What is liberated when citrate is converted to cis aconitate? So the answer is water. Okay. Although aconitase is a lyase enzyme, water is being involved, but it is not hydrolase, it's still a lyase enzyme. Now the next step, जो isocitrate that was formed changes to alpha ketoglutarate. So your isocitrate is a 6 carbon compound. It is changing into alpha ketoglutarate. It is a 5 carbon compound. So we know that one carbon dioxide has been released. So one carbon released as carbon dioxide. Now the enzyme involved is isocitrate dehydrogenase. This is the mitochondrial isocitrate dehydrogenase. So isocitrate dehydrogenase, it has another isoform, which is your cytoplasmic isocitrate dehydrogenase. So it has an isoform, which is cytoplasmic isocitrate dehydrogenase, and it is a source of NADPH. Okay. Now the reducing equivalent generated, your NAD plus is converted to NADH. So this is an oxidative decarboxylation and not just a simple decarboxylation. Let's have a look in the cycle here in the figure. So this was your isocitrate dehydrogenase, and then this isocitrate dehydrogenase enzyme, it is acting upon isocitrate. Okay, then it leads to the formation of alpha ketoglutarate. Here you can see. Okay, so carbon dioxide is removed, and you can see NADH is formed. NAD plus changing into NADH. Okay, so this is your oxidative decarboxylation. Okay, moving forward to the next step, alpha ketoglutarate, which is a 5 carbon compound, it changes into succinyl CoA. Now succinyl CoA is a 4 carbon compound. So again we know that this is the second oxidative decarboxylation taking place. Okay, and your carbon dioxide is released. So what one carbon is lost as carbon dioxide. Carbon dioxide being liberated, and NAD changes into NADH. Oxidative decarboxylations mean that your oxidation reduction is taking place. Therefore, NAD is changing into NADH, and decarboxylation means carbon dioxide is being lost. So this is also an oxidative decarboxylation, not just simple decarboxylation where carbon dioxide is lost without oxidation reaction taking place. So there is no in those simple decarboxylations, there is no generation of reducing equivalents. But unlike this, this is an oxidative decarboxylation. So here you have NADH produced and carbon dioxide being liberated. Now this is an irreversible step, and the most important vitamin required for oxidative decarboxylation is thiamine. Okay, you have other coenzymes also required, which are like your coenzyme A, that is pantothenic acid, your NAD, that is niacin, then your FAD, that is riboflavin, and then you have lipomide. And then you have thiamine as well. So thiamine is the most important vitamin required for oxidative decarboxylations. Okay. Now alpha ketodehydrogenase is the enzyme which is bringing about this reaction. Alpha ketoglutarate dehydrogenase. This is the enzyme, and we have studied another dehydrogenases or another oxidative decarboxylations where in our previous class, just the previous class that we have done, which was on branch chain amino acids. If you remember, a branched chain keto acids formed from their branch chain amino acids. They were acted upon by the enzyme branch chain keto acid dehydrogenase, if you remember. So carbon dioxide was being lost, and what was being generated? NAD plus was changing into NADH, remember, and acyl co acyl CoA was being formed, which was one carbon less. Okay, and the deficiency, if you remember, or when there was defect in this enzyme, it was causing leading to maple syrup. Okay, so please don't forget what we've already studied. And also another example of oxidative decarboxylation, that is your link reaction. Link reaction where your pyruvate changes into acetyl CoA. This is also an example of oxidative decarboxylation where carbon dioxide is being lost and your NAD plus is changing into NADH, and the enzyme is pyruvate dehydrogenase. So this is one example of oxidative decarboxylation. Second, and the third one being in TCA, your alpha ketoglutarate dehydrogenase and your isocitrate dehydrogenase. Okay. Now, succinyl CoA changes into succinate. This is brought about via the enzyme thiokinase. Okay. GTP is produced only in those organs where there is gluconeogenesis. So what is happening? When your succinyl CoA is changing into succinate, either ATP can be produced or GTP can be produced. It can use both ADP and GDP. So depending upon which organ it is taking place, if it is taking place in one of those organs where your gluconeogenesis is also taking place, then in that case GTP is produced. So in this step, your succinyl CoA changes to succinate, and either ADP or GDP is converted to ATP or GTP. GTP is produced only in those organs where your gluconeogenesis is taking place. Why? Because if you remember in gluconeogenesis, there was this enzyme called phosphoenol pyruvate carboxykinase, which was changing oxaloacetate into phosphoenol pyruvate, and this step was requiring GTP. Okay. You see, everything is interconnected. You just cannot afford to forget what we have already previously studied. Okay. Now moving on to the next step, that how your succinate is being converted into fumarate. So the enzyme for this reaction is succinate dehydrogenase, which is present in complex 2 of electron transport chain. So we have already spoken about this, that it is present in the inner mitochondrial membrane and not in the matrix. It is present in inner mitochondrial membrane. Okay, and this is the only dehydrogenase which releases FADH2. Okay, now your fumarate changes to malate. The enzyme is fumarase, which is a lyase enzyme. So this enzyme, it catalyzes the addition of water across the double bond of fumarate. So water is added to the double bond of fumarate, and your double bond changes into single bond to form malate. A malate to oxaloacetate. This is brought about by the enzyme malate dehydrogenase. So in the first half, you see the dehydrogenases of the first half, that is your isocitrate dehydrogenase and alpha keto dehydrogenase. They undergo oxidative decarboxylation. We've just discussed what's that. And the dehydrogenases of the second half, that is your succinate dehydrogenase and malate dehydrogenase. They are simple dehydrogenases, okay? So there is only oxidation reduction reaction taking place, but there is no liberation of carbon dioxide molecules, okay? Here, let's have a look in the cycle. See how your alpha ketoglutarate, which was formed here, then this is acted upon by the enzyme alpha ketoglutarate dehydrogenase to form your succinyl-CoA. Now, this succinyl-CoA is acted upon by succinate thiokinase to form your succinate. And in this step, you see one ATP is being formed. This is your substrate level phosphorylation. That ATP is formed at the level of the substrate only and does not have to enter electron transport chain. Now, this succinate changes into fumarate. The enzyme involved is succinate dehydrogenase. And here FADH2 is being formed. Now, this fumarate, you can see the double bond of fumarate. Here, this is the double bond fumarate. To this, water is added via the enzyme fumarase, and you can see malate is formed. The double bond has changed into single bond. Then this malate is acted upon by the enzyme malate dehydrogenase, and NADH is formed and leading to the regeneration of oxaloacetate. So that was about TCA. Okay. Let's have a look at the MCQs. Which of the following is not an intermediate of TCA? The options being acetyl-CoA, citrate, succinyl-CoA, and alpha-ketoglutarate dehydrogenase. So we see citrate is an intermediate, succinyl-CoA is an intermediate, alpha-ketoglutarate is an intermediate. So answer being acetyl-CoA. Okay, acetyl-CoA is not an intermediate of TCA cycle. Acetyl-CoA and oxaloacetate, they are the starting materials. They are the starting materials, okay, the starting substrates. And not the intermediates. Okay, the first substrate of Krebs cycle is, your answer being pyruvate, glycine, HCl, and lipoprotein. So your options are these. Your answer will be pyruvate. Inhibitors of TCA. Fluoroacetate, it non-competitively inhibits equinase. Then arsenite, it inhibits alpha-ketoglutarate dehydrogenase. And your malonate, it inhibits succinate dehydrogenase. Okay, here in this cycle, we can see these inhibitors. See, this is your fluoroacetate inhibiting equinase. Then you have your arsenite. This brings us back to our clinical case scenario. Okay, so arsenic poisoning, it will inhibit what? To alpha ketoglutarate dehydrogenase. And your malonate, it inhibits your succinate dehydrogenase. Okay. So these are the inhibitors of TCA cycle. Now moving on to the energetics of TCA. 3 NADPH which are produced. 1 NADPH gives 2.5 ATP. So 3 NADPH will give 2.5 into 3, which is 7.5 ATP. Now 1 FADH was produced. 1 FADH gives in the electron transport chain 1.5 ATP. And there was one substrate level of phosphorylation, so 1 ATP being produced. If you add all these up, you get 10 ATP. So per TCA cycle, that is 1 TCA cycle gives you 10 ATPs. Okay. You can see the steps where NADH, 3 NADH, 2 in oxidative decarboxylation. Here, one NADH, second NADH, and then by malate dehydrogenase. So 3 NADH and this FADH2. So one FADH2 and this one ATP. So this forms the energetics. Now, one MCQ we have here. High energy phosphate is not produced in the options being TCA, hexomonophosphate pathway, glycolysis, and beta oxidation of fatty acids. So we've already studied this, that your HMP was involved in the formation of NADPH and in formation of pentose sugars, okay, for DNA and RNA synthesis. It had no role in ATP or energy production. So your answer will be HMP. Now moving on to the significance of TCA. So acetyl-CoA is completely oxidized. So one of the significance of TCA or the importance of TCA is complete oxidation of acetyl-CoA. The other is it is an amphibolic pathway. What is an amphibolic pathway? That it is has both catabolic and anabolic roles to play. So the catabolic role of TCA is acetyl-CoA being completely oxidized, and the anabolic role being citrate can form fatty acids, so fatty acid synthesis. The other being your alpha ketoglutarate can form glutamate, and this forms GABA. So if you remember the amino acid chapters that we've already done, your alpha ketoglutarate can form glutamate. How? By a transamination reaction. Okay, so alpha ketoglutarate can be transaminated to glutamate, and then your glutamate on decarboxylation will form GABA. So glutamate on decarboxylation forms GABA. So this is one significance or anabolic role of TCA, that is formation of GABA. Then your succinyl-CoA forms heme. In heme synthesis, we will study the entire pathway of heme. But in glycine also, I had already told you how succinyl-CoA plus glycine, they combine and then undergo a series of reactions to ultimately end up forming heme. So that is how your heme is being formed. And then your oxaloacetate forms glucose. That was via gluconeogenesis. Okay. Now, another very important significance of your TCA is anaplerotic role. That is filling up reactions. Let's have a look at what is this. So anaplerotic reactions are replenishment replenishment of depleted intermediates of TCA. Okay. So let's have a look at this. Your citrate. Your citrate here, it can form what? It can form fatty acid. Okay, it can go for fatty acid synthesis, and therefore it can get depleted. Then your alpha ketoglutarate, it can form glutamate as we have already studied, and then that glutamate can form GABA. So, and therefore your alpha ketoglutarate can get depleted. Then your succinyl-CoA, it can go for heme synthesis, and therefore it can get depleted. And then your oxaloacetate, it can go for glucose formation, and then it can get depleted. So these intermediates that are being depleted from the TCA, it is very important that they are replenished. Otherwise, what will happen? Your TCA will be affected. Okay, depletion of intermediates of your TCA, it will lead to a non-functional TCA. Okay, depletion of intermediates of TCA, it can lead to a non-functional TCA. Your TCA will be impacted if the intermediates, obviously, if the intermediates are depleted, then how will the cycle run? It will be affected. So the intermediates, they need to be replenished after they are used for anabolic roles. So anaplerotic role is replenishment of these depleted intermediates of TCA cycle. That is why it is known as filling up reactions. You fill up the depleted intermediates that have been used for various anabolic purposes. So how is it brought about? You can see pyruvate to oxaloacetate. Your pyruvate is a 3-carbon compound to oxaloacetate. This is a carboxylation reaction, and this is one of the most important anaplerotic reaction. Okay, so that is one way your pyruvate, it gives oxaloacetate and therefore has it forms one of the most important anaplerotic reactions. Then the other is your alpha ketoglutarate. You can get them from histidine, arginine, proline, orchin, fatty acids, and glutamate. Then your succinyl CoA, it is replenished by valine, isoleucine, and methionine, which forms propionyl CoA, and then propionyl CoA ultimately forms succinyl CoA. And then your fumarate, it is replenished by phenylalanine and tyrosine. If you remember the catabolism of phenylalanine and tyrosine, ultimately we were getting fumarate, and then your oxaloacetate is replenished by aspartate. Okay, so this was about the filling up reactions or anaplerotic reactions. Now, hyperammonemia inhibits TCA cycle by depleting what? So our answer will be alpha ketoglutarate. Okay, we've already studied this. How? Because your alpha ketoglutarate, it was forming what? Glutamate. Okay, when there was hyperammonemia, your alpha ketoglutarate was being depleted. Now, moving on to the regulation of TCA. So the regulatory enzymes of TCA are your citrate synthase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and pyruvate dehydrogenase. Now remember that pyruvate dehydrogenase is the link reaction. It is not part of TCA, but it is involved in regulation of TCA. So it regulates TCA, but it is not part of TCA. So if you get an MCQ that which of the following enzymes is not part of TCA and you have these 4 options, your answer will be pyruvate dehydrogenase. Okay, this is the link reaction and it is the link between the TCA and the glycolysis and not considered as a part of TCA. It is only involved in the regulation of TCA. Now, how high energy inhibits TCA cycle. Okay, so the high energy, when will be TCA more active in you? When the body needs ATP, because TCA is basically involved in ATP production. So when there's already high energy in the body, high energy state of the body, it inhibits TCA. Your hypoxia, that also inhibits TCA. Now, high ATP ADP ratio inhibits TCA because if there is high energy ATP, your TCA is inhibited. High NADH. NADH also is ultimately, if there's high NADH, it will go into ETC, electron transport chain, and will form ATP. So high NADH and NAD ratio also inhibits TCA. So isocitrate dehydrogenase is inhibited by NADH and ATP. All dehydrogenases in TCA are favored by increased level of calcium in muscles. Okay, so if the calcium level is increased, what will happen? All the dehydrogenases are stimulated or activated. They are favored. The activity is enhanced by calcium. Now, in the brain, TCA is regulated at the pyruvate dehydrogenase level. So PDH is the regulatory step of TCA in brain. PDH is the regulatory step of TCA in brain. No, there is no hormonal regulation for TCA. Okay, this is important. So TCA should have taken place whether the body is in fed state or the fasting state. So there is no insulin or glucagon regulation or TCA. Okay, it takes place whether the body is in fed or fast state and there is no hormonal regulation for TCA. Okay, so with this we come to the end of our lecture. Thanks for watching. If you have any queries, you can send them in.